safe research data is very important, and how we have to manage the data, how we collect the data, and what is the importance of data collecting, what are the ethics, rules, and regulations. So safety is the first priority in any research. So let me share the PowerPoint. Okay. So safe research means regarding the data, this had four constituents, basically 4 points. First thing is data management, confidentiality of data, ownership of the data, and ethics. So I will go one by one in detail. So data management means it's a process of validating the data, organizing the data, collecting the data, protecting the data, maintaining the data, processing it to ensure the accessibility of the data, reliability of the data, and quality of the data. So data management comprises of 10 things basically, and throughout the lecture these 10 things will be repeated in different aspects. So there may be redundancy of some of the terms. So first thing is data management plan, ensuring data quality, consisting naming conventions, then documenting data thoroughly, securing storage, controlling the access of the data, adhering to the ethics and the legal standard of the institution where we are working, backing up the data, then the version control and the training of the personals. And after following these 10 things, when we are collecting the data, analyzing or publishing anything, it helps us in maintaining the scientific rigor of the data and the research, using the data consistently and accurately. That means if somebody is living in between and next person is joining, he or she can use the same data in the same way which previous person was using and accurately. And using the data by more people in the research area other than the person who is working on it. Means if you are publishing it, the same data set can be used by other persons also. So I will go one by one into the detail of each of this. So the data management plan means we should have a plan before starting the research, that how we will collect the data, what data will be collected, how we will store it, how we will manage it, who will analyze it, what data is needed for the particular hypothesis to prove that. It's not necessary that if we are going to the medical records of the patients or in the basic research, we need to collect everything, whatever we are seeing there. So we should be very rationalized in collecting the data, that what data we need to prove our hypothesis. Then data quality, we should always check for the quality of the data, whether the data which we are collecting is accurate or not, whether it is complete or not, and the consistency. Like if we are collecting for the clinical research, if we are collecting the patient CMP and BMP, or we are collecting the age, BMI, height, all parameters. So whatever the patient population is, that we have to look for that each and every patient which we are including into the research should have that data. And if some patients are not having, some patients are having, that means it's not consistent. And we can drop the patient which do not have the data which we need. Next thing is consistent naming and file organization. This is very important, because if we are collecting a lot of data or tons of the data, that means we should organize it properly. Otherwise, it will be a problem when we will be analyzing the data at a later time point, or maybe after 3 months, 4 months, 5 months, then it will be tough to look at what data we collected. So whenever we are collecting the data, it should be properly named, organized in different files, either year-wise or month-wise or day-wise, depending on the project. Proper naming should be there on each and every file. Documentation. Documentation is very important in research. So whether either you are doing a clinical research or basic research, Whenever we are collecting the data, we should always document that what day we have collected it, what data we have collected it, what was the method used to collect that data. So each and everything should be documented and co-signed by a person who is witnessing you when you are collecting that data. Secure storage. That is another important thing. Whatever the data we are collecting, whether in a clinical research or basic research, we should secure the data and store the data in a place where no one else can have the access. The access should be only to those persons who are involved in that research. Other than that, we should not give the access to anyone. Plus, another important thing is storing the data in a place where you will not lose it. Sometimes computer may not work, or something, it may got viral infection or something, it got corrupted or whatever has happened, and you are not able to get the data from your computer. So we should storage data some other places also. There is a rule of 3-2-1. 3-2-1 means you should have a 3 copy of the data. You should store in 2 places, and 1 place should be offsite. Like if you are working in the lab, you can store the data in your lab computer, then you can store the data in your office computer. Plus, you should have the data in one external disk also, so that in case the online data which you are collecting in the lab or in the office, you are not able to access that data due to any reason, you have the data in your external disk. So there should be 3 copies, 2 places minimum, and 1 more places should be offsite. Then access control, as I discussed earlier, who should have the access should be determined before starting the research. These are the persons who will collect the data as well as access the data. So there's a difference between collecting and accessing the data. Like in a lab, postdoc or research tech, they are collecting the data also. And PI is not always there to collect the data, but PI have the access. So this should be determined before starting the research, that who will collect the data and who will have the access of the data. Next thing is legal and ethical standard. So while working with the data, or collecting the data, or analyzing the data, we should adhere to the legal and ethical standard of the institution. So whatever the rules and regulations are there in either university or any company or any institution, we should follow the rules, maybe related to the data privacy, intellectual property, or research integrity. That who data belong to whom. If you are filing a patent with the data, who will have the ownership of that? Who has the intellectual property of that patent? Then data sharing, with whom you can share the data. Obviously, the person involved in the research, you can share the data with them. Then if you are publishing that data, then if somebody is asking for that data, then you have to share the data if the grant is federally funded. If you are using your own grant, or if it is a university grant, or a company grant, then you don't need to share the data publicly. Means publicly availability of the data is not needed. But if it is a federal grant or DoD grant, then it is our responsibility to have the data publicly available. If it is not federal funded, then the funding agency has the full access of the data. You have to share the data with them. Along with that, we can share the data with the person who you are collaborating with. So these things should be in the written policy before starting the research. Version control means you should keep the data in different versions. Means first we are collecting the raw data, that is version 1. And when you are working on that data, mining the data, or working on the data, analyzing the data, there may be some changes, because you may be removing the outliers, you may be doing some other statistics. So that will be version 2. And then when you are analyzing using GraphPad Prism or SPSS or any other software, or if you are doing bioinformatics of the data, then data may change. Not the basic data will change, but the outcome may change. So that each version of the data should be given a different name, and it should be monitored that which data is what, and it should be properly organized. Then training. Training means educating the person who are working in the research, that how to collect data, what data need to be collected, where to store the data, whom to share, whom to give the access to the data, how to analyze the data. Each and every person who is involved in the research should be trained. And this is the responsibility of the principal investigator to train each and every person who is working in the research. Then data security means you should keep the data in such a place where data is secure. Means it should be secured by either encryption or by password protection. Like if you are, for example, if you are storing the data in your lab computer and it's a common login ID, then each and every person can have the access to that. So either you should have a encrypted data or you should take the data out from the common computer and keep the data in your computer where it is password protected, or keep it in a external drive which is always encrypted. So security is very important. Related to do the deletion of the data. Deletion of the data means when you can delete the data. So any federal funded research or DoD research or any research which is funded by federal agencies or DoD or any company, it is mandatory to keep the data for 5 years after the completion of the research. We cannot delete any data point or anything which is related to the research till 5 years. After 5 years, it depends on PI whether to delete the data or not. If they want to keep the data, they can keep it. If they want to delete it, they can delete it. Sometimes, if it is a very high impact manuscript, it takes time to publish, and you may need the data even after 5 years. If somebody is asking a question on your paper that this thing is not clear in the paper and you need to provide some raw data, then you may need that data. So it is important to keep the data with the PI. You may delete it, but the data which is not needed, it's all up to the PI whether to delete it or not. The next thing is confidentiality of the data. So some of the terms again will be redundant, and so confidentiality of the data comprises of few things depending on the whether you are doing a clinical research or you are doing a basic research. So I will be discussing both. If you are doing a clinical research, then first and foremost important thing is informed consent. You should take informed consent from each and every patient in such a manner that they understand what is written on their informed consent. And they should sign it only when they understand it. That what you are going to with the patient, what data you are collecting about the patient, whether you are going to use that data only for analysis, or if you are collecting any blood or any tissue, are you going to do any genetic analysis or not? And if you are doing any genetic analysis, the counselor should be there. So you should explain each and everything to the patient that what data you will be collecting, what tissue you will be collecting, what blood sample you will be collecting, what you will be doing with that samples or the data which you are collecting. Only after that, the patient should sign the informed consent. Second thing is data collection method. So data collection method means what is the various ways or what are the various ways by using them you will be collecting the data, whether you will be using the medical record or you will be using the blood samples and will do the analysis on the blood samples, or you will be using some tissue for multiple wet lab experiments or your omics analysis or anything else like metabolomics, proteomics. So each and everything should be written before starting the research that what will be the outline or what will be the pathway to work on the data which you are collecting or tissue you are collecting or blood samples, whatever you are collecting from the patient. Same thing in the basic research. Whenever you are starting a project, you should define that what are the various experiments you are going to do. Whether you are going to do only staining or you are going to do only immunostaining or RT-PCR for gene analysis or Western blot and ELISA for protein estimation or some advanced techniques. So each and everything should be written that what are the various techniques you are going to use for the data collection. That's called a data collection method. Secure data storage, again, as I discussed earlier, each and every data, whatever you are collecting or getting after experiment, should be stored in a secure place. Then ethics, use of data. So this means that the data which you have collected should follow, whenever you are using it, should follow the rules and regulation of the institution where you are working. You cannot use that data for your personal use. If you are collecting a data from the patient. And if you are looking at that data and you found something interesting, you can share that thing only with the person who are involved in that research. You cannot use or share that data with someone else in your family or friends. Like one example is if you are working with the animals, taking photos and making the videos of animals with whom you are working are not allowed. And if you are taking any photo or video with the permission of ICAC or IRB, even then you cannot share those photos or videos with your friends. They are only for your research purpose. You cannot put on the social media, you cannot put on your emails anywhere. So it is strict, and we should follow the ethics and regulations in that case. Same with the clinical research. If you are doing the collecting the data from the patient, we have to follow the HIPAA policy. We cannot deviate from HIPAA policy. We have to respect it. Then limited access to the data, as I discussed earlier, who can access the data should be delineated. Everybody should know that who can access it. Then team education. Each and every person should be taught, should be aware of confidentiality of the data, that how to make the data confidential, what are the rules and regulations, how to store it, how to secure it, how to use it. So there are few components for elaboration: informed consent and data collection. So obtain the informed consent. As I discussed earlier, we should obtain fully informed consent. Like you can see here, it's written fully informed, means patient should be informed of each and everything before taking the consent and getting the consent signed by the patient, that how the data will be used. Data collection method, choose an appropriate method and minimize the risk of revealing the data or sensitive information to the community. Establishment and data management process, that how the data will be collected, where it will be stored, and all these things. Next is secure data storage and handling. So limited access, I discussed. Then secure storage, you can store in three places, three copies, two places, one is offsite. Then password protection, data should be stored in a place where you can use a password and nobody should know that password other than you. Then secure file transfer. This is very important because sometimes if you are collaborating with different universities or different PIs are collaborating, they share data with each other. So the data which you are sorry, sharing with each other will go through email or via Dropbox. So the file transfer which you are doing between the two PI or between the two university or within the university, it should be secure. Either they should be encrypted or secured by a password. Then coding and anonymity. Like if you are collecting the data from patient population, this applies to clinical research. So you should have a coding like patient number 1, 2, 3, 4, 5, 6. We cannot take the name of the patient in the data, because at the later time point, even if you are leaving the university or you are leaving and someone else is working on that data, and if they say, okay, this patient is this much, this is the name, this is the date you collected, this is the age, so they can track that patient back, that okay, this is the patient living in this area. This should never happen. You should have some coding, patient number XXYZ or something like that, and then only the PI who is collecting the data. Like if you want to do the intervention in the patient after the research, then we need the identifier of the patient because we have to intervene. So in that case, the person who is doing the analysis, working on the data, publishing it, should not know any identifier. So only the clinician who is giving you the data or who is working with the patient should know the identification of the patient, that patient number is such and such, patient number 2 is such and such. So that in case we need to intervene in the patient, then we know which patient we have to call for again. De-identification means we should not collect any identifier like age, name, telephone number, social security number, address, these things. We can collect age, BMI, height, race, those things, sex, but we cannot collect any identifier. for our research purpose. So de-identify is very important, and personally identifying information should not be collected. Encryption, as I discussed earlier, each and every data should be encrypted if you are working with the human patient. Even in the basic research also, many universities has a rule that your flash drive, whatever you are using, or external hard disk, whatever you are using, should be encrypted. Now coming to the ethical use and the disposal of the data. So ethical use means we have to follow the rules and regulations of the university. In case of clinical research, we have to follow the HIPAA rule, and in basic research, we have to follow the rules of the university. Like if you are using animals, we have to follow the rules of ICA. If you are working with the human patient, we have to follow the rules of IRB, Institutional Review Board. ICA works for animals. Then data sharing, as I discussed, we have to share data only with the person who are involved in the research, not with anyone else. Disposal means up to 5 years we have to keep the data, and after that we may discard the data, or we can keep the data as long as you need it. Then training and education, again, regarding the ethics and confidentiality, both each and every person in the team should be trained. That these are the rules and regulations about the data to make it confidential. Next thing is, let's see this. Compliance. This is correct slide. Yeah. So regulatory compliance means you should understand the legal and law and regulations of the university, which the legal department of the university will tell you that what you have to follow. And there are the trainings for that, like city trainings. So we have to follow each and every city training. Without that, we cannot work in the lab or with the animals. So we have to follow that. Next, implement necessary security measures like using a password or encrypting the data. Maintain confidentiality throughout the project. As I discussed, each and every data point should be kept confidential. Then anonymization and pseudo-anonymization means no identifier in the data. It should be anonymized. Even if in a clinical setting, if a nurse or a student is collecting, or a postdoc or a medical student is collecting, or a resident is collecting the data, it should be anonymized. No one should know from which patient data is coming from, other than the clinician or the PI. Then next thing is ownership of the data. So for ownership means who owns the data. So it's a power as well as your power to use the data, how to control the data, as well as the responsibility also. Means who is responsible for the data security, management, analysis, and who has the control over data, and really who is the owner of the data. Basically, the ownership of the data goes to the institution where you are working. The PI are the managers. They can manage the data, they can collect the data, they can store the data, they can use and analyze the data. But the data is owned by the university or the institution you are working in. So ownership refers to both the possession as well as the responsibility. It has a power as well as a control. The control and responsibility of the data is held by the institution and the funding agency who is funding you for the research. PI is the manager. It's not the owner of the data. Though each PI has the data, but real ownership goes to the institution or the funding agency. So ownership of the data, the rules and regulations dictate that how the data will be managed, how it will be shared, how it will be protected, and how you will follow the compliance. So these things are predetermined, and each and every research should have these things in writing before starting the research. So it comprises of legal and control and responsibility. As I discussed earlier, your legal of the university will tell you, and you should go through all city trainings to learn each and everything about this, that how we are responsible for data security and confidentiality. Data management and security, same thing, the compliance, we have to follow all the rules and regulations. whether you are in clinical research or in basic research. Then that data stewardship means managership. PI is the manager of the data, who owns the data also and analyze, keep the control of the data. Then data sharing, as I discussed earlier, it should be confidential. It should be shared only with the person who are involved in the research. It should be shared in an encrypted way or password protected way. Then data use agreement means if you are a research scholar or you are a PI and you are sharing the data with someone else who is a co-PI or working with you in the same research, maybe in the same university or in a different institution. Then there should be an agreement between you both that how the data will be used, which part of the data will be used and which part of the data cannot be used, and how that data will be used. Means it will be used only for the publication, or it may be used for filing the patent, or it may be used for further developing a new drug. So each and everything should be in written document that how the data will be used in between two parties. Then retention and disposal. Again, this should be written that how the data will be, how long data will be retained, and when data will be, how and when data will be disposed of. Whether it will be deleted or it will be kept in an archive folder in an external drive, and it will be with the PI or with the person who is sharing the data with the PI. Then intellectual property. Again, the intellectual property is the rules and regulations from the university. It may belong to the PI or it may belong to the university. Like our university has a rule of 50-50%, like 50% intellectual property is of university and 50% is of the PI. So each university has a different rule. For the companies or industries, the intellectual property always goes with the company ownership. It never goes with the person who is working on the research. So academia, it is 50-50, and institution means industries, it is 100% of the industry which you are working in. So it should be predefined. Then data sharing policies should always be written down, and to whom it will be shared and how it will be shared, what amount, what is the extent of the sharing, all those things. Then clear communication. So for each of these aspects, each and everything should be clearly communicated, not only communicated via email or via text or via paper format. It should be in a written document signed by both the parties or each and everyone who is involved in the research that we have agreed to these terms and conditions about the data collection, management, analysis, publication, intellectual property, and storage, disposal, all these aspects. It should be clearly communicated between each and every party, and each and every person should sign it that we will follow these rules. And it should be done before starting the research. Coming to the ethics means we have to follow the rules and regulation of the university. Regarding the clinical research, we have to follow the HIPAA policy that we will not misuse the data. We will collect only that data which is needed. We will not use that data as an identifier of the patient. No one else will know about the patient other than the person involved in the research. No identifier will be collected. One and most important thing in the clinical research is genetic aspect. If you are collecting the data for genetic aspect, are you doing only the gene expression level or the protein level or the metabolomics level? Or you are doing the genetic analysis for the pedigree analysis. Means you are looking for his or her previous generations, that how they may be. You are doing the pedigree analysis for the patient. So it should be delineated before starting that what you are going to do with the data which you are collecting. If you are doing any genetic research where you may know, okay, this patient's relative or offspring or previous generation were having this disease or may have this disease due to the anomaly of such and such gene or mutation which you are getting after the analysis. So this is called a genetic analysis, and it is highly regulated. We cannot do without informing the patient. There are rules and regulations. We have to follow that. And there should always be a genetic counselor, that why this genetic study is needed, why we are doing it, what will be the risk, what are the benefits. So each and everything we have to tell to the patient. Coming to the basic research, if we are working with the animals, whether we are collecting the data or the samples from the animal for our basic research to prove the hypothesis, or we are looking for their evolution or origin of the research. Like if you are working with the dinosaurs, fossils, then you are looking into the history. So that should be defined, that we are looking for the history, evolution, how they were evaluated. We will be looking for the various fossils. But if you are working with the swine or rat or sheep, rabbit, hamster, guinea pig, then you are collecting the tissues or blood or any fluid from the animal for your basic research. Then it should be defined that you are going to do such and such experiments. You are not going to look for their genetic aspect. Like, for example, if a swine is getting weight, so why is a specific species of swine or rat or mouse or any animal is getting weight? Is there any genetic anomaly? If the research is having the aim for that, yes, you can do it, but it should be predefined. But all of a sudden, we cannot define that, okay, this animal was on high-fat diet and they are getting weight. So you can look for the molecular aspect, but you cannot look for the genetic makeup, that why they are gaining more weight compared to others. So if you want to look for the genetic makeup, you have to apply for that with the ICAC, that this is a new thing which I have found in this research, and I want to look for the genetic makeup of this such and such animals. Then you have to get the permission from the ICAC to start the genetic research. So there are rules for each and everything. So the research ethics matters for protecting the participants' safety, to maintaining the scientific integrity, upholding the human right and dignity, ensuring social responsibility, building trust in the research and the institution, and complying the legal and regulatory compliance. So these all things belong to the clinical research. So in clinical research, if you are doing something and by chance or by mistake of someone who is involved in the research, data is leaked, and some general public came to know that, okay, this has happened and this is the patient living in this house who has such and such issue like HIV or AIDS or some other diseases which can be transmitted in the general public. So this is wrong. We cannot reveal any data to anyone. So if such thing happens, your reputation as a researcher as well as an institution will be lost. No one will be enrolled in your future research. They will have the same thing. Okay, your data is getting leaked. So why we should roll myself in the research? So you will lose your reputation. The confidentiality issue. So it should be maintained. So this is very important to follow the rules and regulations and ethics of the institution while you are doing a clinical research. Same thing applies to the basic research because here you are using the animals. In animals also, you have to follow the same rules. No photographs, no video, other than if they are not allowed, if they are allowed by the ICAC. And the ICAC will allow you to take the photographs and animal or videos only related to the research. You want to publish them or you want to teach students regarding that. So for teaching purpose and publishing, it is allowed, but it should be kept confidential. So key ethical considerations are taking the informed consent, confidentiality and anonymization of the data minimization. We have to collect the data which is needed. We should not collect all the data. Then data security, fairness and equity. So fairness equity means the data, whatever you are collecting, the output of that data should be equally divided in the community, and the risk and benefit ratio should be looked for that. You should be very fair that these are the risks, these are the benefits. And whatever the benefits you will be getting, like if you are using a patient samples, and for example, if you are using the patient who are responding to a treatment and the patient who are not responding to a treatment. So, and you are getting the answer that these patients who are not responding is due to this reason. No, non-responsiveness is due to this reason. So if they are not responding or responsiveness is due to a mutation, and you get a drug to treat that, that drug should be equally and publicly available. It's not that you will be giving that drug only to the person who are involved in the study. It should be publicly available to each and everyone. So that means fairness and equity of the research and the data. Coming to the transparency and accountability, you should be always transparent. If someone needs the data and related to the research, you should be transparent to show the data to that. You should not keep anything hidden that, okay, this patient or this research is something outlier. I cannot use it. So if you are not using the data point, you should have a rationale for that. And if somebody asks you who is involved in that research, you should reveal it or you should tell it. You should not keep anything hidden within the group. Then accountability means you are responsible for that. You are accountable means if a data is leaked or somebody is misusing the data, then you will be responsible for that, that how that person is misusing the data when data belongs to you. So you are accountable for each and everything, means the person who is doing the research, the PI, the principal investigator, is responsible for and accountable for each and every wrongdoing with the data if it is happening. So each and everything, that's why each and everything should be in written. Everybody should follow it. Training should be given to each and every person who is involved in the research that how we have to handle the data for a safe research. Compliance and regulation, as I discussed earlier, we have to follow as per the whatever the compliance and regulations are there in the university. Best practices for safe and ethical research data is develop a clear management plan, then consult with the expert. Consult with the expert means if you are new to investigation, if you are new to research, and you don't know anything about the data management, data planning, how to collect the data, you should consult with the expert. Means the person who is in the research, who knows how to do all these things, who has a good experience. For legal things, we can go to our legal department. That okay, this is the problem, and this is I am going to do. What should I do? For the basic research, you should meet the PI or the postdoc or the lab manager that, okay, this is the experiment. I am going to do these and these wet lab experiments. What are the rules and regulations to use these instruments? What are the rules and regulations for analysis of the data? What softwares I need to use it? So all those things you should always ask if you don't know. Then train the researchers. It applies to everyone, whether it is a postdoc or a technician or any lab manager or any assistant professional, associate professor, each level it is important that they should be trained for rules and regulations. It is the duty of the principal investigator to train each and everyone. Like for ICAC in our university, ICAC chair is the responsible person to teach and educate each and every student about the city training, that they should do city training before appearing coming to the lab. They cannot touch any animal or any tissue without doing the city training. They cannot even enter into the lab without city training. So those things should be the part of teaching. And each and every person who are doing research, whether basic or clinical, should go through those trainings. Then regular review and update. This means that if a project is of long duration, like for 5 years, 6 years, you should update your data at each time point. It is not necessary that you are getting all the information, like if your patient population is of 100 patient or 10,000 patients, depending on the patient population. So it's not necessary that you will get all the patient at the same time point. You may get those patient data over the years. So you should update the data whenever you are getting the new data in the same file. That's why organization of the data is important, naming an organization, so that you can update that file and review it. Maybe you get the data, you ask your postdoc or your technician or your student to enter the data in the file. And by mistake, it was wrongly entered. So you should review it. That okay, each and every data, whatever we are getting, is correctly entered into the file or folder where we are storing the data. There is no discrepancy in the data. Whatever you are getting, you are entering it. We are human, error always happens, so we should always review it and update it with the date. Like this data was collected on this date and added on this date. Sometimes samples comes, for example, samples comes from the hospital to the basic research lab, and you are on vacation, and your technician is collecting the samples, or lab manager or anyone else in the lab who is present is collecting the samples on your behalf. And they are keeping the samples in liquid nitrogen or -80 or -20. And you are coming back after 10 days, and then you are entering that, okay, I received the samples from such and such patient, patient number 10, patient number 11. This is the sample. These are the two sample, three sample, what a type of sample, whether this is a intestine or skin or cardiac sample or arterial sample or blood sample, whatever the type of sample is. So each and every particular we have to write down. So what date we got the sample and what date you entered the data on the Excel sheet. It should be mentioned. So review and update is very important. Coming to promptly address the issue. So promptly address the issue means if there is an issue with the data collection or data sharing, storage, or analysis, you have to promptly intervene. And this is the responsibility of the principal investigator. For example, if you are getting the blood samples from a hospital and you saw that whenever you are getting the blood sample, they are clotted. So there are many reasons for that, and you don't need clotted blood. So either they are not properly collected in the proper tubings, or the temperature was not maintained during transportation, or it was delayed, the transportation was delayed. So you have to intervene. You have to talk to the person who is collecting, who is transporting, who is receiving, and what time period is there, and if there is a delay, why there was a delay. So each and everything you should check. Coming to the basic research, if you are getting a data from RT-PCR or Western blot or immunostaining or basic staining, and if you see that some values are abnormal, means whatever you are expecting, they are not like that. You have to intervene that why you are getting such wrong values or totally absurd values. So that is called an intervention, timely intervention. So sometimes it happens, for example, if you are preparing a RNA and cDNA and contamination is there in your cDNA or RNA. Contamination may be due to ethanol, propanol, what you are using, triazole you are using, then you are using the DNA RNA free water. So any of this thing, all your hands, the gloves you are using, the pipette tips, the pipette, any of the thing may contaminate your samples. So if that contamination is happening, your data will be totally different. Second reason may be the wrong method. Maybe you are using the primers which are wrongly designed, or the antibody which is wrong, the species is wrong. So there may be thousands of reasons behind the wrong data. As soon as you are seeing that and you are observing that this is not as per the expectations, this should not be the data which you are expecting. You should look back from step one to step which you are, that where it went wrong, and you should intervene to correct it. You should not overlook it. Okay, maybe the next time we will get it. No. First correct it, why you are getting wrong. Then only move to the next step. If you are moving to the next step without correcting it, there are the probability that the next step will always be wrong. You will get the wrong data in the next step. So your whole research will be compromised. To avoid that, you should step in at the first step. That okay, this is wrong, we have to redo it. So that's why we say that either it is clinical research or basic research. Troubleshooting takes over 90% of the time. If we will troubleshoot something at the earliest point, the rest of the research will go very smoothly. So we should troubleshoot each and everything if you are experiencing any issue. Without that, we should not move. So timely intervention is very important. Okay, so this was it for today. And Now coming to your questions. Any question now? No question? Miriam, any question?
No question. Thank you, Dr. I.
V? No. Tony? I think he's not there. Felicia?
No questions. Thank you.
Okay. So if no question, Natalie will be sharing this PowerPoint with all of you. And you can email me anytime if you have any question regarding the data management for the safe research. Okay. Thank you.